So let's dive into gene expression. How is gene expression controlled? Gene expression has to be controlled tightly in our system, right? Now, in one of the chapters, I don't remember which from the top of my head, I did tell you that a gene is actually, say, let me draw a segment of DNA here, and let me actually draw a gene in that segment of DNA. So here is the gene. Let's say this is the gene for insulin. I'm just saying anything, right? So the gene starts here and it ends here. Now, oops, that's not what I wanted to do. Now, let's say that in that gene, we have a segment right here. That is the promoter. And a segment right here, that is the terminator. In the middle, right here, we have the open reading frame. That open reading frame is what is going to become the protein, right? So we have a promoter, an open reading frame, and a terminator. I want you to remember that. The reason why I want you to remember that, it's because for a gene to be expressed, a promoter is going to play a very important role. You have some transcriptional factors that come and bind on the promoter, right? And this transcriptional factor, upon binding on the promoter, they are going to pull RNA polymerase that is going to come and do transcription, right? So the integrity of the promoter sequence is essential in gene expression, because if a regulator comes and bind somewhere here, it can shut off gene expression, right? But by the same token, if a regulator comes and bind, I'm going to change my color here, comes and binds somewhere here, this can cause actually a change in the conformation of DNA in such a way that the RNA polymerase is going to be more likely and attach more easily on that promoter, causing an activation of gene expression, right? So you have transcriptional factors that are going to play a very important role. And this transcriptional factor are going to bind on the promoter sequence, right? Or before the promoter sequence. So they're going to either activate gene expression or shut off gene expression, all right? Then you are going to have possibilities of extra copies of genes. That extra copies of genes is going to make sure that if one gene is not expressed, the other one is going to be expressed, right? Okay? So these are some levels of control of gene expression. Now, let's dive into some more. The structure of the genetic information, in other words, how the genetic information is packed in the nucleus, plays an essential role in gene expression. The most effective way gene expression is going to be controlled, it's before that gene is transcribed. Let me repeat that. The most effective way a gene is going to be expressed, it's before that gene is transcribed. In other words, the cell is going to play with the structure of chromatin, what we call chromatin remodeling, that is going to either expose some segment of DNA that need to be expressed or not expose some segments of DNA that do not need to be expressed, so that exposure of DNA or not exposure is going to determine the expression of genes or not. And one gene can be exposed in one particular cell while not exposed in another type of cell. So chromatin remodeling is going to work as an on and off switch. Another dimension is going to be the microRNA. The microRNA are small RNA molecules that are actually going to come and bind to prevent gene expression or to prevent translation. All right? We are going to talk about that. And finally, you have small segments of proteins, what we call microproteins. These microproteins actually can bind on proteins or can bind on the gene so that the gene is not expressed or the protein is not active, right? There are many levels at which gene expression can be controlled. So here we have a transcriptional factor. We have three molecules of transcriptional factors. These transcriptional factors can physically bind on the promoter to interfere with gene expression, but they can also bind away from the promoter to stimulate gene expression. All right? Two, you have microRNAs. These microRNAs are going to recognize some portion of the messenger RNA so that translation never takes place, right? Actually, in the control of gene expression, you have a transcriptional control of gene expression, right? You have a post-transcriptional control of gene expression. Then you have a translational control of gene expression and post-translational control of gene expression. So it's a lot of information, but we are going to unclutter all of that. So the transcriptional factor play a very important role in chromatin remodeling. But not only transcriptional factors, but also other aspects that we are going to take a look at. Now, what are some of the elements that play a role in chromatin modeling? Now, remember, DNA in human cells is never naked. It's complexed with proteins, and remember, these proteins are called histones. These histones play a major role in gene expression. How so? By the simple nature of being attached to DNA. Because remember, in gene expression, for the RNA polymerase to come and transcribe the gene, the two strands of DNA have to open up, right? The two strands of DNA have to open up, all right? Once the two strands of DNA are opened up, the template strand is going to be read so that a messenger RNA can be produced. All of that implies that there is no protein like histone in the neighborhood that is going to cause some steric issues, right? So that DNA has to be naked for a gene to be expressed. But histones are always around, right? Which means that in the chromatin remodeling, there are some instances where histones have to move, right? But the question is, what causes the move or not of these histone molecules, right? So, for these histone molecules to move, few things can happen to them. One is acetylation, the addition of an acetyl group that is CH3CO2. All right? So we have acetylation of histone, we have methylation of histones, and we have phosphorylation of histones. Now let's see how these different modification of histones are affecting gene expression. Acetylation and phosphorylation both are going to work toward activating gene expression. In other words, acetylation and acetylation and phosphorylation are going to be such that the histone molecules are going to change their conformation so that they free up DNA, and the RNA polymerase and the transcriptional factor can come and bind on that free DNA molecule. That's one. So they work for turning genes on. That's one. Two, the methylation. The methylation actually do the opposite action of acetylation and phosphorylation. How so? Whenever a methyl group is attached to histone, this is going to cause the gene expression to be turned off. In other words, histone is going to be even more attached to DNA so that there is no room for RNA polymerase, there is no room for transcriptional factors to stimulate gene expression. Now, let's take a look at some graphic representation here. So remember that chromatin is DNA and histones, and this DNA and histones are organized in nucleosomes. So this is one nucleosome, this is another nucleosome, another nucleosome, another nucleosome. You have some pieces of DNA right here that are exposed, right? Which we can say, okay, well, this is going to be some DNA that can be read so that a messenger RNA is produced from that segment, except that this segment are just too small to house a whole gene, right? And some of our genes are huge, all right? So the question is really, how do we get this DNA to be free? Well, and this is when we go back to what I said earlier. So let's take a look at acetylation shift, okay? Acetylation binding. That acetylation, okay? So now we have a gene that is initially turned off, okay? That gene that is initially turned off is going to go through the following sequence of events. One, you have an enantiosome, right? That is right here, okay? That enantiosome is going to house an enzyme that is going to be complex with an enzyme that is called acetylase, right? And in as much as that enantiosome is combined with the acetylase, we are going to have this complex of histone that is formed right here and blocking the TATA, the Tata box, right? The Tata box is blocked. And notice that we have 4 chains of histones. We have 2 chains of H4 and 2 chains of H3. And you have some tails of each of the chains that are exposing, right? This is when the gene is turned off. But now, when the gene is turning on, right? What happens is that first of all, a signal has to come and tell the cell we want the gene to be expressed, right? And once this is said, then what happens is that there is a large complex protein that comes, binds on the enantiosome, which is actually going to, interfere with its activity. But along with that, what we see is that some small proteins are going to cause the actually, I'm sorry, let me modify something here. So you have an acetylation, right? An acetylation that is going to happen in this histone, this one, and this one. These acetyl groups are causing, along with the binding of the large protein complex, right, are going to cause the unfolding of this whole structure. Now, that unfolding occurs because of the change in the conformation of the histone proteins upon attachment of acetyl groups. Okay? So that change in the conformation is going to cause the histones to move away from the Tata box. And as the histone move away from the Tata box, now there is room for the RNA polymerase to come and attach and do its job, right? Okay? So now we do see 2 forms of chromatins, right? We have a chromatin here that is actually compact, and we have some chromatin here that is loose, right? So the chromatin that is loose is going to cause exposure of some pieces of DNA where genes are located, right? That exposed DNA carries a gene right here. Now that gene can be expressed, okay? However, the gene that is right here cannot be expressed, okay? It cannot be expressed because of the steric, the cluttering caused by all these histones here, okay? So about 2% of the chromatin in our body is open and easy to read. Everything else is cluttered and hard to read. Now, you can think that in a muscle cell, for instance, some genes find themselves in these areas, and these genes will never be expressed in as much as they are in that area, while other genes are going to find themselves, let me change color here. Let me rather take this one. There we go. So you can find a gene that is right here, okay? Right here, right here, right here. And all these genes are going to be expressed, okay? And in that muscle cell, this gene will be systematically exposed so that actin filaments, actin proteins can be produced, myosin proteins can be produced, and myosin and actin proteins are the proteins that are responsible for, the contraction in the muscle. All right. How about the microRNA? The microRNAs are non-coding RNAs. Non-coding RNAs mean they don't actually lead into protein formation, right? There is no protein equivalent for these RNAs. These RNAs are very short. They are about 21 to 22 base long, which is not a lot, right? Then, when we look at our genome, there are at least 2,555 microRNA. These microRNA are going to regulate at least one third of the proteins and the protein encoding genes in our genome. Okay, so one third, it's not nothing, it's a lot. Okay, so a typical human cell is going to have about 1,000 to 200,000 microRNA molecules, right? This speaks to the role of this microRNA in gene expression. In other words, what the cell is trying to do here as a strategy, it's if if the cell did not get successful at controlling transcription, they are going to at least control translation with the participation of the microRNAs. All right? So the binding of microRNA to a target messenger RNA is going to interfere with translation. And by interfering with translation, there is not going to be any protein, right? That is basically what I mentioned earlier by saying post-transcriptional regulation. All right. So microRNA can be very much used in medicine, including in the treatment of cancer. We found that certain microRNA are more or less abundant in some cancers than in healthy individuals. The same thing is observed in people with concussion, right? Where actually there are a number of microRNA that are actually transported by some vesicles that are called exosomes. So these exosomes are going to leave the cell to actually actively go out of the cell. Okay? And it has been shown that in case of concussion, right, 15 microRNAs are going to differ in levels in the cerebrospinal fluid between people with concussion and people who actually do not have concussion, right? So that shows that, well, in these individuals, right, if we can target this microRNA, right, this can be a benefit to, dealing with concussion. All right, now the microprotein. Microproteins are very small protein. This small protein have less than 100 amino acids, right? They can actually fit in other molecules, but in other proteins, so they can bind to a cell surface protein such as a receptor protein, an ion channel, right? And by binding to these proteins, they are going to interfere with their function, right? And this is what we call post-translational regulation, right? But post-translational regulation is much more complex than what I'm just saying. We're going to see that a little later. So genes that actually code microproteins are going to exist within other genes. At least that's what was shown, right? So microproteins are not just part of a larger proteins, but they are their own segment of DNA that produce a small segment of messenger RNA that is going to actually produce these microproteins. Okay? One type of microproteins are going to be the microproteins that actually tag messenger RNA for destruction. Now, let me take an example. Say you are hungry, you go and get yourself a sandwich, and you are full, and you eat on a regular basis, and you actually need some enzymes for digesting this food that you are eating. Then at some point, you decide to undergo starvation. Okay? Now, as you undergo starvation, there are some genes that will not need to be expressed at that point, right? But remember earlier, everything was fine. You were just eating normally, right? So you had a certain pattern of gene expression at that time. When you undergo starvation, you have a different pattern of gene expression. Now, all the transcripts from when everything was fine, all this transcript, something needs to happen with this transcript, right? So that they are replaced by transcripts compatible with starvation. Now, the microproteins are going to tag the transcript from when you were healthy and causing this transcript to be destroyed. Okay? So that's an example, real example, right? Now we are going to dive into how is the genome organized. Now, in terms of genome organization, I'm not going to really dive into the sequence of alignment of genes more than we are going to look into how many genes do we have for how many proteins, for how many transcripts, right? And try to relate all of that. Now, when we look at the entire human genome contains about 20,325 genes. Okay? This 20,325 genes are going to encode about 100,000 messenger RNA. That's a huge difference between the number of genes and the number of messenger RNAs. One can wonder, wait a minute, the extra messenger RNAs, where are they coming from? Right? Now, eventually, we are now going from more than 100,000 messenger RNA to more than 1,000,000 proteins. So, wait a minute. We are going from roughly 20,000 genes to roughly 100,000 messenger RNA molecules to 1,000,000 proteins. How do we go to this considering the central dogma, right? Now, we are finding out that the cell has a way of doing things that actually make all of this possible. All right? Now, what are some of these approaches that make a cell to actually have more protein than they do have genes. We have some examples are alternative splicing, the use of intron as actually coding information, which normally is not, right? We have protein modification after the production of the protein, and the cutting of precursor proteins into two or more proteins. Okay, so let's dive into this. All right, so during gene expression, we have that DNA is transcribed into RNA. So we go from DNA to RNA. The RNA is going to be, first of all, modified in the nucleus. And from there, that RNA is going to come in the cytoplasm. And now that RNA is going to be subject to a number of things. All right. Now, what are some of the things? We're going to have microRNA that can block protein synthesis. We are going to have protein folding that can happen not properly. We can have polypeptide shortening that can happen. We can have sugars that can be added. We can have polypeptides that can aggregate together, right? Now let me kind of go back in the nucleus. In the nucleus, as RNA is produced, we know that the initial RNA that is produced, the pre-messenger RNA, right, is not necessarily the messenger RNA. Alternative splicing is going to play a major role in the number of different RNAs that are produced. Chromatin remodeling is going to determine which genes are being expressed when with which intensity, right? The promoters are going to account for gene expression. So when it comes to gene expression, there are really a lot of different things that contribute in gene expression. And the highest level of gene expression being at a DNA level, right? Being at a DNA level, okay? Is DNA being available for gene transcription? Yes or no, right? Is the promoter properly structured? Yes or no? Is there a mutation there? Yes or no? Even if it's available, right? Then the messenger RNA, is it matured? Yes or no? And how? Right? Okay. So now, in terms of maximizing gene expression, we are going to see the role of exons. These are the coding portions of the gene in producing multiple types of proteins from one exon. From one gene. Okay. Alternative splicing is the one that actually allows different combinations of exons. Now, a gene in a human is going to be a piece of DNA, right? That piece of DNA actually does not contain all parts that are coding, right? Actually, let me modify what I'm saying. What I'm trying to say is that a DNA for a given gene does not contain exclusively coding portions. It contains coding portions, right? Exon A, exon B, exon C, and exon D, and non-coding portions, intron 1, intron 2, intron 3. All right? Upon transcription, you are going to have a pre-messenger that is produced. That pre-messenger is going to undergo alternative splicing. That alternative splicing is going to produce mature messenger RNA that can be made up of exon A, B, and D. In the case of ABD, right? It can be made up exon A, C, and D. It can be made up of exons A, B, C, D. At the end of the day, all these proteins are different. From one single gene, we have 3 proteins shown here. But guess what? We could have much more, all right? But let's move on. This actually shows you the power of alternative splicing in producing a wide diversity of proteins. 2, it's from 2 genes. You can have 2 genes out of one single gene. One gene, you have 2 genes. How can that happen? Well, in this particular instance, you have in DNA again, you're going to have a whole sequence, right? And that whole sequence is going to have exons and introns, okay? This is an exon right here, another one here, another one here, another one here, and another one here, okay? So exon 1, 2, 3, 4, 5, all right? So, and then we have introns, 1, 2, 3, 4, right? In this particular instance, we are going to address the gene that is responsible for the production of prostate-specific antigen. Now, that's prostate-specific antigen is going to actually have 4 introns, okay? 4 introns and 5 exons. So we have 1, 2, 3, 4, 5 exons. Together, when the splicing is done, we have the PSA protein, right? However, that same gene can be expressed in such a way that exon number 1 of PSA, prostate-specific antigen, is going to actually combine not with any other exon, but actually with intron 4, in such a way that intron 4 for the PSA-linked molecule represents an exon. So this one is an exon. For the PSA-linked molecule. Please remember that, right? So that the PSA-linked molecule, it's one gene that is completely not the PSA gene. All right? Understand that, okay? So from one single gene, you can have actually 2 genes. And what we see is that the 2 genes, when they express their products, are going to work in an antagonistic way. In other words, the prostate-specific antigen that is abundant in someone who has who is at risk of developing a prostate cancer, right, is going so while PSA is going to be abundant in someone who is at risk of developing prostate cancer or has prostate cancer, the LM is going to be present in small amount. Likewise, LM is present in high amount in a healthy individual, while PSA is present in smaller amount, right? So and they kind of work against each other. All right. So now, introns can be used as alternate genes. Let me explain. So when we consider a particular gene, and that gene, it's right here, okay? That gene has two exons, exon 1 and exon 2. And that gene actually encodes a protein that is called neurofibromin. That neurofibromin is going to have two exons, okay? So when the gene is expressed, you're going to have splicing, and that splicing is going to bring exon A and exon B together to produce neurofibromin. However, that DNA molecule that has this intron as intron, for the opposite strand, right? For the opposite strand, so the non-coding strand, the intron in here is going to actually carry genes in the other strand, so that we have another gene here, and another gene here, and another gene here. So what was an intron in one context is many genes or an exon in another context. So that's another way of actually multiplying the possibility for many proteins more than the number of genes that are on the chromosome. For post-translational modification. The post-translational modification can occur after the protein is produced. After the protein is produced, some residues can be added to that protein. These residues can be sugars, in which case we're going to call that protein a glycoprotein. These residues can be lipids, in which case we're going to call the protein a lipoproteins. Okay? Glycoproteins and lipoproteins are very much found as surface markers of cells. Okay? These surface markers of cells are going to show the specificity for a particular tissue, a particular organ, or a particular individual. Right? Some cell surface markers are essential to skin cells that are different from those that are found on the surface of white blood cells, different from the one that are found on the surface of liver cells, and so on and so forth. Finally, but not least, you have the cutting of the final product. Okay? What I mean by the cutting of the final product, it's that a given gene, when converted into a transcript, is going to produce a protein, and that protein, it's actually not the final product. That protein has to be cut into two to form two different proteins with two different functions, yet they come from one single gene. Now, this is the case of a particular health condition that we call dentinogenesis imperfecta. In dentinogenesis imperfecta, you have two proteins that are absent. We have DPP and DSP. DPP and DSP are produced from DSPP gene. That DSPP gene, it's the dentin silophosphoramidase protein. Let me repeat that. Dentin sialophosphoprotein. The dentin sialophosphoprotein, it's the gene, right, that encode that whole long protein, right? Then we have a messenger RNA for DSPP, and then we have the precursor DSP and DPP proteins. Okay? So that precursor is going to be cut right here so that we have the production of the dentin phosphoprotein and the dentin sialoprotein. All right? Now, the dentin phosphoprotein is very abundant, while the DSP is present in low amount. Okay? So these proteins play a very important role in the integrity of the dentine in the tooth. Under the NML, right, you're going to have the structure of the tooth where the DPP and the DSP are going to go and play a role. In the absence of this, right, the tooth is very soft, right, and actually breaks apart. All right, so what are the mechanisms that maximize the genetic information? We are going to have alternative splicing. We're going to have introns encoding one isoform is an exon in another, right? We have an intron in one DNA strand can be an exon in another DNA strand, right? We have a post-translational modification of the protein, and these are two. One is the addition of a sugar to form glycoprotein, the addition of lipids to form lipoproteins, and finally, last post-translational modification is the production of a precursor protein that is going to be cut to actually produce two proteins at the end of the day. All right. Now, when we consider the whole genome, we notice that altogether, only 1.5% of the whole genome encode for proteins. We're going to wonder, okay, what is the need for the rest? The rest actually account for a number of other things, including viral DNA, non-coding RNA, introns, promoters, and other DNA sequence that control gene expression, and also repeated DNA sequences, right? Now, we may think that most of the genome that is transcribed, right? No, let me modify what I want to say. We know that most of the genome is transcribed, right? Most of the genome is transcribed, even though we only have 1.5% of the human genome that encode protein, but most of the genome is transcribed, right? However, one can wonder, well, why only 1% of the DNA is converted into protein? 1.5% is converted into protein. We may think that everything else is junk DNA, which actually we used to think that all that DNA was junk DNA. But actually, we are finding that all that DNA, one, it's not junk DNA. Two, The reason why we may think it's junk DNA, it's because we don't have a good understanding of the role of all these DNA sequences, right? But now, with time, we are getting to a better understanding, right? So now we know that while only 1.5% of the human genome account for proteins, okay, we know 8% account for viral DNA, and this viral DNA are DNA that comes from previous infections. Okay, we have some DNA that are replicated, repeated. These are the transposons. Okay, transposons represent about 45% of our genome. They are replicated, they are repeated sequences, right? In our DNA, there are sequences that can move from one place to another. What is the role of these transposons? Well, we are only scratching the surface of transposons in here, but we know that they play a major role, including changing the pattern of gene expression. But let me close that parenthesis. You have some DNA that is going to account for pseudogenes. Okay, so these are pieces of DNA that look like genes, but they are actually not encoding proteins. Okay, we're going to have bunch of introns, represent 26% of introns, right? Simple sequence repeats represent 3%. Okay, segmental duplications of DNA represent 5%. And then we are going to have telomeres, centromeres, and other pieces of genetic information that we actually don't know what they account for, right? We know that they are found in the heterochromatin. Heterochromatin, it's that part of chromatin that is not expressed. While euchromatin, euchromatin is a chromatin that is compatible for gene expression, right? And other that we don't know very well is represent 3.5% of our genome, right? The viral DNA, again, represent about 8%. These are these are there because of past infections, right? Among that DNA that actually come from retroviruses. These are RNA viruses that come and insert in our DNA. Okay? Now, if you consider, for instance, HIV infection, when you get infected, right, you have many people who are infected with HIV that actually live a normal life, even without taking antiretrovirals. Why? Because the HIV virus, it's actually a type of virus that we call a temperate virus. When it inserts, its genetic information goes and insert in our genome and stays silent. That can last throughout your life without ever expressing anything, right? So there are actually bunch of retroviruses that have been now part of our genome that actually are now so much part of our genome that they are completely not foreign anymore, right? So this viral RNAs that are now part of our genome are called human endogenous retroviruses, or HERVs. These HERVs have shown to be actually present in some cases and very characteristic and standing out. In some cases, for instance, in some cases of people with amyloid disease, right? AML, the multiple sclerosis, and myeloma. The herbs have been associated with these diseases. How so? When herbs are overexpressed, we see that in individuals with amylosis, multiple sclerosis, and melanoma, which are cancers, we see an unusual high expression of herbs. All right. So this slide, it's basically listing a lot of the different things that are part of the genome that do not account for proteins in our genomes. All right. So I'm not going to bug you with this list, but it's just to for your information. We have the viral DNA, we have the non-coding RNA genes, we have the transfer RNA genes, ribosomal RNA genes. Or there is one thing that I want to show you, the pseudogenes right here that look like genes, but they are not actually. You have the long non-coding RNA that control gene expression, and so on. You have a bunch of different genes, different parts of the genome that do not account for proteins. So that's about what we are going to cover in this chapter. Thank you for your attention. Until I talk to you again, take care.